We're a research laboratory and marine science education center and shellfish production hatchery located in Beals, Maine, in the heart of Downeast, Maine, in Washington County. We also serve as the marine science field station for the University of Maine at Machias. So today's seminar will last about 40 minutes. And well, the presentation itself will last about 40 minutes, and we're scheduled to end at 1 p.m. So this is a sort of regular Zoom meeting. It's not a webinar format. So I would just like to remind everyone to, when Alex is talking, especially to keep yourselves on mute. After the presentation's over, we're going to take as many questions as possible, keeping in mind the end time of 1 p.m. And you can ask a question by raising your hand on Zoom or by adding it to the chat. And this presentation is being recorded, and we will upload it to YouTube for those people that weren't able to attend today. So just everyone keep that in mind. And I would also like to thank Hannah Green, who is the Downeast Institute's Island Institute Fellow, for helping with the technology and moderation today. So thank you, Hannah. We're really excited to share today's speaker with you. We're going to hear from Alexandria Ambrose on how she is evaluating Maine mussel farms as marine habitat using underwater video. And so Alex is one of DEI's inaugural 2022 Downeast Institute Fellows. And this work that she's presenting on today represents her work that she's undertaking this summer as part of her fellowship at Downeast Institute. So Alex is finishing her first year as a master's student in Rutgers Graduate Oceanography program. Her research interests are in how organisms are using available resources in changing habitats and how those changes can impact an ecosystem. She is applying her research interests to aquaculture, focusing on ecosystem services. And we're really excited to have her present today because as many of you have known, that our wild populations of mussels have suffered greatly with green crab predation that is caused by the 40-year warming trend. Their populations have been able to explode because of the 40-year warming trend in our oceans. So, we're increasingly looking towards farming and husbandry as a way to sustain Maine's mussel production. So this is really good to learn about how other organisms are interacting with the mussel lines. So with that, I will hand it over to Alex to learn more.
Thank you, Sarah. I'm going to share my screen. Okay. As Sarah mentioned, my name is Alex, and most of my research focuses on ecosystem services and specifically habitat provisioning, which I will be talking to you today about, specifically on mussel farms in Maine. So starting kind of broadly, thinking about what habitat is and what is included in habitat. And Oxford Dictionary defines it as the place or environment where a plant or animal naturally or normally lives and grows. And depending on where in the world this is and the type of environment it is, it can look very different, depending on the organisms that are using it and how it's being used. But a lot of habitat tends to have some of the same elements. And one of those elements tends to be structure, specifically 3D objects that will either increase surface area and allow organisms to propagate, grow, provide shelter. They use it in different mating rituals, rearing young, or in nurseries, in those type of situations. So nature tends to be really good at making habitat. And even if nature has not made habitat for a specific reason or in a certain area, animals tend to make their own. And so when looking at terrestrial environments, you have birds that can weave their own nest and make their own habitats. Or in a case where it's human-made or nature-made, you have caves or burrows that bears and foxes can use. Or even insects like bees tend to make their own hives and own structures they use as a home and as a rearing place for their larvae. But in marine systems, they can look a little bit different because marine animals don't always have the same ability to make a lot of their own habitats. They have to use what is available to them. And this can include coral reefs like this one, or hydrothermal vents, or oyster reefs, and marsh grasses or wetlands that provide a lot of those different structures and habitats that a lot of animals use. But recently, due to a lot of changes in the environment due to climate change, along with other things, a lot of these systems are declining, but they also provide a lot of different things for the environment, and that are ecosystem services, and which are essentially the different goods and services that different environments or habitat provide that benefit humans, but also the environment. So ecosystem services tends to be broken down into different categories of provisioning, regulating, that cultural aspect, and supporting services. And although all of these things can work differently, they all tend to have an end goal that goes towards the overall goal of ecosystem services. But with the decline of a lot of the natural habitats, they're not able to provide these same ecosystem services that they used to. And so over time, a lot of these have gone away, and oyster reefs are at less than 10% of the abundance that they used to be globally within bays. Coral reefs are at less than 30% of the area that they used to cover. Kelp forests are suffering from kelp deforestation due to trophic cascades, and one trophic level being taken out and another one becoming dominant within the system. And marsh grasses and wetlands are disappearing due to sea level rises, as well as eutrophication in coastal areas. And all of these things tend to have their own ecosystem services that they provide, but with them declining, all of those services are also disappearing. And so with the disappearance, with those declines, humans have taken it into consideration in trying to recreate a lot of those habitats. And they look differently for all of the instances and situations that they're going into, or the locations that they're going into. So you have coral restorations that are happening. There's a bunch of different methods for oyster reef restoration, as well as wetland or estuary marsh grass restorations. And although all of these are good and they provide those benefits in ecosystem services, they also have potential a few downsides. And upfront, when creating a lot of these restoration areas, they tend to cost a lot of money, if not hundreds of thousands or millions of dollars, depending on where you're putting them and the scale that in which you're putting them in. You have to take into consideration the materials that are being used, where is it being put, how big is it going to be, all of the manpower that goes into it. Also taking into consideration that how you're understanding how it's going to interact with the environment. And all of those things, it also requires mitigation and regulation. And so for these systems to survive long-term, there has to be maintenance involved in it to make sure that they're providing the functions of doing what you put them out there for. And so there is the potential for other structures to provide ecosystem services that don't have some of the caveats of these restoration habitats, and that would be aquaculture. And aquaculture comes in many different forms, shapes, and sizes, and they use a lot of different gear types. But one of the common things about these gear types that tend to tick off a lot of the boxes within restoration tends to be they create those complex structures for organisms to grow and propagate and feed and all of those other things. But there's also minimal extra effort. And by that, I am referring to aquaculture upfront does cost money when a farmer is putting out the lease or applying for their lease and trying to determine the different gears that they're using, the location that they needed to be in. But all of the maintenance is already kind of included in that lease formation in the beginning. There doesn't necessarily have to be extra work done outside of the work that's already being done on the farm to maintain it. And responsible farmers are the ones who are checking on their farms on a semi-regular basis or how often they need to in terms of cultivating what animals that they're growing out. They're making sure that their gears are not destroying or causing too much damage to the areas, and they're not losing a bunch of things that are ending up in the environment around it. So ecosystem services has been studied in aquaculture farms, especially on shellfish farms, because of the different organisms that they include, but also because of these different gear types. And they have the same different categories of provisioning, regulating, habitat, support, as well as those cultural aspects. But for a lot of these things to happen, there's other things that have to be taken into consideration. That includes the farming method, understanding the type of gear that's being put out into the environment and where it's being put out into the environment, understanding where the hydrodynamics of the area, is it in really active areas or high flow or low flow? What does the tidal cycle in that area look like? How the benthos is composed of, what organisms are living in the benthos in the area? How does the biodeposition from the animals that you're putting out there going to impact that? All of those different things need to be considered when thinking about the ecosystem services provided by shellfish aquaculture. So if you look specifically at the different functions that some of these shellfish gears or shellfish farms can provide, there's a lot of different things that they can do, and studies have been done on them. And so in this review, there was 129 papers that provided or estimated ecosystem services that are provided by shellfish aquaculture gear. And some of these might be slightly hard to see, so I'm going to go through them. And you have nutrient removal. Here you have livelihoods in terms of creating jobs. You have habitat provisioning, you have carbon sequestering, augment wild fisheries, you have tourism, water clarity, acidification regulation, and coastal protection. And all of these things are great in the different things that they provide. But specifically for this presentation, I will be talking about habitat provisioning, which refers to a structure being used as habitat, providing elements of habitat in an environment. And they don't always have to be the first function of what you're putting out there. Like, for example, with aquaculture gear. And so you might be asking why aquaculture gear. There might be other different ways that don't cost as much money to kind of still keep these ecosystem services, and that is true. But specifically, aquaculture is growing globally, and as it continues to grow globally, because commercial fishing has declined due to loss of habitat for a lot of the species that they're fishing for, stock declines of overfishing for certain species. And so this is from the NOAA 20th, the latest fisheries report, and over 40% of the value of aquaculture in the United States comes from the Atlantic coast. And so as you begin to expand the footprint of aquaculture in these different areas, there also needs to be a greater understanding of the impact that they're going to have in the environment and what that means for the environment that they're going into. How is it going to interact with the gear? All of those things have to be taken into consideration. And specifically, habitat provisioning has been studied before on shellfish aquaculture farms. In Virginia, they looked at floating gear using lift nets. In Rhode Island, they've looked at cages using drop nets and suction dredges. And although this is great that research is starting in this area, the sampling methods that they're using are known to either be destructive to the environment or the habitat around it, or does not always accurately look at the fish assemblage within that area. And so recently there has been a small shift into using underwater footage to capture this same type of data with minimal direct human impacts, but also allows for the potential to look at a greater assemblage of fish that might be using those types of gears. And studies that have done this include studies from Connecticut that looked at on-bottom cages, as well as in Washington State that looked at on-bottom cages, flip bags, and on-bottom oyster cultures and clam cultures. And once again, all of these are great that these studies are starting to come out, but all of these studies, except for the one, are on oyster farms. And oyster farms, it makes sense because oyster farms are one of the most proliferant aquaculture industries at the moment due to the money value, but also because of the value that they potentially have for the environment. But this leaves a hole in the data in terms of the different types of gears that are used in other common aquaculture gears or shellfish farming, like long lines that are used in mussel farming. So if you're looking at these gear types, these are all used in oyster farming. You have bags with racks, and you have flip bags, and you have off-bottom cages or on-bottom cages, as well as floating bags. And kind of from these pictures, you can see that a lot of this gear tends to use space horizontally because those are typically kind of the easiest to maintain, and also where kind of how oysters tend to use space the most is horizontally within their natural habitats. But long lines used commonly in mussel farming tend to use more of the vertical space in the water column, which has the potential to provide different habitats in different ways compared to oyster farming gear, which is why I am doing this research here at DUAD. And so for this presentation, I will also be including data from a study done in an off-bottom oyster farm in New Jersey, so that you can get a feel for what a full data set will potentially look like. And since I am still in the process of collecting here data in Maine, I will be sharing preliminary results for that. But the objectives of this study were to determine how fish are interacting with those different gear types using underwater video, but also determining how underwater video compares relative to some of the traditional methods that have been used so far. So the study designs for these two places are slightly different. You kind of learn things as you go along. So in New Jersey, opportunistic videos were taken, meaning that data or videos were collected typically whenever the farmer was going out, was working with the farmer in terms of convenience. And so the cameras were put out to collect continuous videos, meaning the cameras were put out and hit record while farm work was happening. And those were collected from July through September. And in Maine, I'm still doing opportunistic camera deployments as well as collecting continuous video, but I'm also collecting interval footage. And so continuous video, again, is when collected while farm work is happening. And interval footage is collected while there's no one present on the farm to answer those questions of what do those fish assemblages potentially look like and how they might be different when there's human presence on the farm versus when they're not. And so interval videos are shorter than continuous videos because they're recorded every hour over to cover a full tidal cycle. So the videos are in 8-minute segments that happen every hour from whatever the time point was set at, to cover more time and to look at a longer point in the day compared to the work that is happening on the farm that happens maybe within an hour or 2 hours. So the farm locations for both of these in New Jersey is located in Barnegat Bay, which is more of like a marshy habitat. There's marsh grasses nearby, and kind of like a small sand embankment. And in New Jersey, it's on the southern side of Acadia National Park, which tends to be more of a rocky coastal environment, and the water depth goes to about 80 feet within this area. So video sampling was done using GoPro cameras, and specifically in New Jersey, the gear types that were studied were floating bags which rise and fall with the tides, so the oysters are essentially never exposed during or out of the water at any point in time, and off-bottom oyster cages, which refers to the cage itself is sitting on the bottom, but the bags that are inside the cage and the oysters in the cage are never actually touching bottom. And these have the potential to be exposed at low tide, so the oysters would be out of the water for some point in time. But cameras were attached to the bottom of the floating bags and to either the top or the sides of the off-bottom cages. In Maine, my setup is slightly different. And so due to the environment looking different. So the control in New Jersey was a marsh edge. And in in Maine, the closest replicate to that of this mussel longline would be a natural kelp forest. And so to kind of shift to make it more feasible for the area, we decided to do an experimental gradient design, which looks at the impact at the raft and how that impact changes as you move away from the raft. And so you have images that were collected from inside the raft at raft edge and as you move further away. Another thing that was different in Maine compared to New Jersey is that because mussel longlines use that depth of the water column, I'm looking at 2 different depths. And so collected video at shallower depths at around 2.5 to 3 feet to about 14.5 to 15 feet. And so this is what the farm looks like. It uses 40 by 40 foot rafts, and the mussel lines suspend about 40 or 50 feet down into the water column. Another thing that we're doing different is using supplemental data in terms of minnows. And so potentially to catch smaller fish species to determine how they're interacting with that gear. And so gut content analysis will be done on those organisms. And this is just a closer up image so you can kind of see the layout of what the design looks like. Videos were analyzed using BORIS, which stands for Behavior Observation Research Interactive Software. And this is a free computer software that allows me to catalog and categorize the different behaviors that are being observed. And so how this works is the videos are input into BORIS and you play them in the center, and you have your species and you have your behaviors. The species you can put in as you're watching the videos, or you can add species that you expect to see prior to watching the videos, and the different behaviors that might be associated with that, whether it's foraging, hunting, mating practices, all of those different things. And so you input the keys for both of the species and for the behavior that you're looking at, and it comes out with a certain timestamp. And all of these, all of the data can be extracted into an Excel file that can easily be transferred and manipulated to do all of the data analysis that you need. This is not necessarily an AI device where the software can identify what species you're looking at. It just makes it easier for the person watching it to be able to catalog and categorize what they're actually seeing. So before going on to the results, I'm just going to show you some of the videos that have been collected for New Jersey and Maine. We're going to start in Maine, in New Jersey. And some of these are hard to see, so I'm going to point my laser pointer in the direction of where they are. This one is a lot harder to see, but that's due to the functional deficient wanting to be. The feathered bunnies are here. It's kind of hard to see them with the biofouling that happens on these different gear types. Here you have a sheephead that you can see is foraging on the biofouling that is grown on these gears. As well as a punter that is inside of the cage foraging on the biofouling as well. Here you have a heavily fouled spider crab that is checking out the cage. We have a blue crab, which are really common in New Jersey, that is attached to the underside of a floating bag, which is why it is upside down. And you can also see grass shrimp and a lot of the other fouling that occurs on some of these. Have a juvenile gray snapper, you have mummy chogs, as well as silver sides, and all of these small brown dots as you see on the bottom are mud snails. You can also see how shallow this water is compared to potentially what the waters look like in Maine and the habitats that are used typically in mussel farming. You have your sheephead over in the corner, as well as some silver perch that will be coming up. Have a school of mullet that check out the floating bag on their way by. Have a mummy chog once again, and this one is kind of hard to see, but there was a skillet fish coming up here in the center. We'll move on to some of the videos that I've collected so far from Maine. This video is not as long, but so far the biodiversity has not been as large. So you have a printer that is going to appear here in the corner, as you can see is foraging on fouling that is occurring on this mussel line here. Have a lion's mane jellyfish at the shallower depths. The clip beforehand was taken at a deeper depth, around 15 feet. I have a rock funnel that kind of appears here in the corner. This video was also taken at a deeper depth, about 15 feet. There are no species ID on it. It's more about looking at the behaviors observed. So you have a fish here and a fish that comes in. We also have another fish that's come around and is here. And he swims immediately down when he sees the other fish. Moving on to the results. And so we're going to start with New Jersey. In total, 247 videos were collected, which equated to about 42 hours of footage, and almost 9,000 observations of individual organisms were observed over the 3 habitats of floating bag, off-bottom cages, and a natural marsh edge that occurred on 7 sampling days from July through September. And there were 19 species that were observed and these species were deemed to know or to have to exhibit habitat preference compared to some of the other species that were not included in this. So looking at all of these species individually, I'm going to walk you through this because this is a lot. And so max-in was our measurement of abundance that is on the y-axis, and that refers to the maximum number of fish per segment, 1 minute segment of video. And this allows us to reduce the potential for recounting fish that might swim in and out of a frame multiple times within one video. On your x-axis, you have the different video dates, and it's broken up by the different habitat types. So cage, floating bag, and marsh. And from this, you can tell that there are definitely some species that appear throughout most of the videos. You can also see a small seasonal change in some of them, like your Atlantic silversides, your mummy chogs. And also some species that we're definitely seeing more at one habitat type compared to another. So you have higher max-in of silversides at the cages, as well as for Atlantic silversides, which tend to Atlantic needlefish, which feed on silversides. And there are some that literally only appear at one habitat type, like your silver perch, your kingfish, your cunner. And these made an appearance maybe once or twice, just possibly on their trek through migration to wherever their final destination was within this habitat. So looking at all of the numbers of individuals and kind of looking at a broader scale outside of species, there are more individuals observed using the cages or that were associated with the cages compared to the floating bags in the marshes. And you can also see that there is a small seasonal trend as you start to go into the fall season. There is a decline in total for all habitats for the number of species. The number of individuals that you saw. But when looking at species richness, there is no discernible correlation in how the number of species that use either of the different year types over all of the sampling days. And these are 2 visual representations of a non-metric multidimensional scaling, which is essentially a type of ordination techniques that allow you to look at the correlation between points. And so points that are closer together tend to have higher correlation compared to points that are further apart that have a lower correlation. So the data put into this was the total number of individuals, so that the total number of individuals, which was the sum of the max-in. And so if you're looking at the different treatment types or habitat types, there's small groupings that happen within the cages that correlate with the graph that I previously showed you. There's also small grouping that happens within the floating bags, but there's still some correlating points that don't really correlate as well as these. And for the marsh habitat, there seems to be less correlation between the number of individuals saw there. If you change that to look at the month, it's the same where the number of individuals were put in. There's not that same correlation between the time of year or the month and when those number of individuals appeared. And you have small groupings, but there's not as strong correlations as you see within the different habitat types. We're going to move on to my preliminary results for Maine. As I said, I'm still collecting footage. And so this is what I have collected so far and kind of what I've seen so far. And so I've collected 172 videos over 3 sampling days in June and July. And I've been able to watch 63 of these videos so far, and have observed 319 observations of animals. And 5 species have been identified so far, being punter, lionhead jellyfish, lion mane jellyfish, rock gunnel, moon jelly, and comb jelly. Just kind of looking at the distribution of the videos that I've collected, I have more videos of continuous footage compared to interval, because interval videos require almost replicate or close farm days happening together. And I have not been able to do that just yet, but before I finish my fellowship, I will definitely be collecting more interval footage. And then looking at the locations, I've collected videos from inside the raft as well at the raft edge. And there's slightly more at the inside of the raft compared to the raft edge, but I'm going to finish collecting more raft edge videos as well as more videos that occur at a distance away from the farm. So just kind of looking at basic observations that I've seen so far from the data, over 60% of the observations that I've looked at have occurred at those deeper depths of around 15 feet compared to the shallower depths. And no observations have been made at the raft edge compared to actually looking inside the raft. I've also haven't caught anything in the minopots, so nothing, no gut content analysis has been done yet. But I've observed some of the different behaviors that have mostly been associated with certain species. And so in that video I showed you, you could see that the punter was foraging directly on the mussel line. And then I've also seen behaviors of following or chasing that I've showed you briefly. I'm going to show you more of a distinct video. And so you have a fish here, and this is a punter. That fish comes back and chases the punter around the mussel lines. He does another loop until the punter eventually loses the fish that is chasing it. I have not yet been able to identify the fish that is chasing the punter, but once I'm able to do that, hopefully I can identify what this chasing behavior actually means. Moving to some of the discussion points for New Jersey first, a lot of the species that were seen were associated with either one or more than 2 aquaculture gears. And so this has also been seen before in a study that looked at cages and a made reef habitat. And so they had a higher abundance at their cages compared to the reefs. And this is done in Delaware, in the Delaware Bay area. But they also saw more number of juvenile species similar to what we have seen in New Jersey videos. And so there's potential that the oyster farm itself is creating more foraging habitat compared to what the natural or the restored reef habitat was at that point in time. As I mentioned, we saw a lot of juveniles, but we also saw a year of young and a lot of different adult species, which might suggest that the gear type is being used as sort of a nursery function and providing that different structure for these fish to get bigger, while also providing enough food sources for them to stay. And this might suggest that even with the decline of natural oyster reefs within the area, because a lot of these species are solely associated with oyster reefs or have been known to be highly correlated or observed using oyster reefs as habitat, that the gear might provide some habitats that, or the sources of habitat that natural reefs might be lacking. Moving on to just some of the discussion points and kind of what I'm thinking through for the data in Maine. Punter, I expected to see punter because punter is a common species here, and they're known to be associated with structure, kind of like you would see in this video, the punter swimming through the lines, kind of going on about his business, potentially foraging on some of these lines. So that was fairly expected to be seen. And then rock gunnel tend to be associated with habitats that have denser algal cover or tend to pick habitat both based off of vertical depth, which I've also been able to observe. And all of the all of the rock gunnel that I have observed have been at those shallower depths, or about 2 feet, where the algal fouling tends to be more heavy due to the light source being brighter. And again, with this chasing behavior, until I am able to identify sexually what this fish is, I'm not going to be able to determine what the behavior means. So whether it's aggression or a mating ritual, or if it's predatory, it's possible that this could be a much more mature cunner that is just pushing around smaller cunner, because cunner are known to be aggressive towards other fish, other cunner, as well as humans. And so I'm still working on that to hopefully discern what that behavior exactly means. Just some main takeaways. So when looking at this type of data, it is the term like oyster, shellfish gear, as well as mussel shellfish gear can be used as habitat elements, provide those elements of habitat in different ways, even in different locations, even if they are the same species like cunner. And also there's the potential for these farm structures to be providing more elements of habitat than some of the surrounding declining natural habitats due to various reasons. The data from the data from New Jersey has been published, and you can receive that paper here using this QR code. If not, I can also send or email the paper to you if you are interested. And my next steps are I'm finishing out my fellowship here at DEI in the next few weeks. And so my goal is to finish collecting those videos to kind of fill in some of those gaps of questions about what those assemblages look like when there's farm work happening, when there's not farm work happening, to have more discernible correlation between the different depths, but also the locations, looking at the different gradient as you move away from the farm. Just some brief acknowledgements. Thank you to all of the New Jersey funders for that project, as well as DEI for funding me for being here for the summer, as well as the owners of the farm back in New Jersey, as well as the owner for the mussel farm here in Maine. And just a quick plug for next week's seminar is by our very own, our DEI's own, Brian Gill and Robert Holmberg, about using shellfish hash for buffering mudflat acidification, which is really big right now as clam farming and numbers are changing due to different climate change reasons. So make sure you tune in for that. And with that, I will take any questions that you have.
Thank you, Alex. That was a really interesting with a lot of information presentation. And if people have a question, please raise your hand, or if you're in the conference room, Hannah, help me see if someone has their hand raised in the conference room. Well, Alex, could you explain how this research fits into like the larger research that's going on with National Marine Fisheries Services, NOAA?
Yeah, absolutely. So the data that has been collected from New Jersey, as well as other institutions, is being headed or being used to create tools to help in the leasing process. And so the project itself, or the program itself, is being headed by the NOAA Milford Lab that have also been collecting this type of data. And it includes institutions like Rutgers, Northeastern University, universities in Connecticut that are collecting habitat provisioning data using underwater video on different aquaculture gears. And so they've been in contact with different leasing regulators and local regulators in terms of helping shellfish farmers obtaining leases in areas that they may not have potentially been able to before, or taking that consideration of that ecosystem service into consideration when giving out leases.
Great. I think Brian had a question.
Alex, can you hear me okay?
Yes, I can.
Great. Thank you for the presentation. It's a great job. I've got a question about either the size or the age or the length of time that the shellfish may have been in the water. So when I look at the mussel lines of Evan Young's in Frenchman Bay, those lines look pretty well established, like they've been there for a while, because I couldn't really make out much of the shape and the density of the mussels, but I could see things like kelp. and other associated epifauna and epiflora on the mussels. So my question is, this habitat provisioning, has anyone taken a look at the question of how long does it take for the habitat to become provisioned? Or if there's an age in terms of the shellfish or their size, if that matters in terms of what you might have seen in New Jersey in terms of oysters. I mean, when those oysters go in the bag, they're, let's say less than an inch, and within 18 months, they're ready to harvest. And so, not only are they changing in size, but they're changing in the amount of water that they're filtering. The bags have been out for longer, so at the end of 18 months, there's a lot more organisms growing on those bags. I'm just wondering about time and aquaculture.
That is a really good question. Here in Maine, I'm not sure how long they've been out. My guess is the video that I've collected, those lines have been out for over at least a year and a half, or at least a year. And so that's why the fouling tends to look a lot more profound, or the buildup of mussel feces on the deeper depths of those lines. But in terms of New Jersey, I'm not really sure if the age or the size of the mussel plays as big of a part as it might in the mussel long lines. And so a lot of the structure that comes from the mussel long lines are from the mussels themselves, because they're attaching to those lines. And so they're the ones actually making those complex structures, compared to in the gears that are used in the shellfish, in oyster farming. The structure tends to come more from the gear that's being used itself. And so those cages, the bags, every now and then in New Jersey videos, you can see a fish that might have swam into a bag that was small enough and got happy enough because there was enough food and never decided to leave. And so it got too big to leave at some point. But I'm not 100% sure, but that would be a good consideration or to look into as data continues to be collected, at if the size matters in terms of the shellfish itself, and how that plays a role into the habitat provisioning.
Great, thank you. I also want to point out that one of the slides that you showed, you referenced Merengue 2010.
Yes.
Yeah. Well, Frank was an undergrad student at the University of Maine at Machias.
Oh, well, like full circle.
Yeah.
We have a question in the chat. Wonderful talk, Alex. Question: are there folks working on applying AI or machine learning to identifying animals and behavior in videos like this?
I know that there is AI software being developed for these types of reasons, to be able to identify different organisms in different habitats, but I'm not sure if they've been used in marine or aquatic systems yet. And so although you can say that a fish looks this way, depending on the angle, the color of the water, all of those different things that go into what a fish looks like and the quality of whatever that image or video is, the AI might pick it up as something different. So I don't know if the software is developed far enough for that to happen in terms of marine systems, but I'm also not very familiar with how well they work in terrestrial systems either, although the clarity would be due to camera quality problems and not the environment quality problems.
And we have another question from the chat. This is from Carissa, and she asks, do you have any control sites in Maine?
So rather than doing control and impact, so as I mentioned, the closest control for the mussel lines would be a kelp forest. And so trying to find that and kind of figure out the best time to put out cameras. And also it's a really high time for lobster buoys at the moment. And so it would be too much conflict potentially to put out those type of lines in kelp forest. And so what we decided to do was that gradient design. And so you're looking at the impact that the farm has at that point, and then as you move further away from the farm. And so the videos were collected inside the farm or the mussel lines, and then videos have been collected at the edge, so not in the farm, but maybe about 15 or 20 feet away from the farm. And then I'll be continuing to collect videos at a further distance away from that, so you can see how the change happens from the source of the impact as you move away from the impact.
Any other questions for Alex? All right, since there's no other questions, I think we can, we'll end the seminar a little bit early and thank you, Alex, for your, for this really interesting and informative talk today.
Thank you for having me.
Yes, and please, everyone, our next, as Alex mentioned, our next seminar will be with Dr. Brian Beal and Dr. Robert Holmberg next week on Tuesday at the same time. And we're looking at how applying oyster shell hash impacts bivalve recruitment, commercially important bivalve recruitment, and carbonate chemistry of the mudflats. So thank you, everyone, for coming today. Really appreciate it.